On the Preprocess tab, we calculate chemical properties and other meta information that we want to append to the molecular identification file. At this point, we've assumed you've successfully uploaded your data into FREDA and optionally defined groups for your samples. If you have not done this, please see the video about uploading your data into FREDA. On the left, we have checkboxes indicating which values and chemical properties you would like to calculate. There are hover icons giving information about each property, and this information is also contained in the glossary. By default, element ratios and Kendrick mass and defect are selected. These are required to produce Van Crevelen plots in the case of element ratios, and Kendrick plots. We'll select some extra values we want to calculate. Here, Gibbs free energy, double bond equivalent, and elemental composition, and hit process data to calculate these properties. We'll choose to review our results and see that FREDA produces table summaries of all our numeric variables and our categorical variables. Here we can see we've calculated elemental composition. Additionally, you can see histograms and bar charts of all values that were originally included in your molecular identification file or calculated here. These values can be used in subsequent tabs, such as the Filter tab. For example, if you wanted to filter out peaks that were in some certain range of hydrogen to carbon ratio, or if you wanted to color a plot based on some metadata value. At this point, we can move on to either quality control, filtering, or visualization. Please see subsequent videos for how to operate these tabs.